Oh, so everyone virtually was not hearing me. Sorry about that. I didn't know. So where was I? Okay, so the HTML report at the end would show you all the QC things that you've done and what happened to the data. But also you would have other stats file and analog file. So if you wanna look more closer into the things that you don't understand about the data, you can actually do that as well. But now I want to quickly compare the results I get from scPipe to the popular CellRanger workflow results. So just to briefly tell you how the data was generated, it was generated in the lab where equal mixture of nuclei from 5 cancer cell lines are collected, and then they were when the pre-processing happens, we actually do separate it into 1:4 ratio and do the processing separately for those for 80% and the 20% gems. But the thing here is that we actually know the ground truth about this data, so we can use that information to do the clustering and validate things downstream. And just quickly show you the time and memory requirements that we have. So basically, in the worst case scenario, this would take up to 2 hours for the 20% data. And oh yeah, I forgot to mention that I will be showing information about the 20% data set for the interest of time, right? So as I said, 2 hours, but that means that if you haven't still made the genome index, or if you decide to do the remove duplicates within the function, within the package, but otherwise it would take less than 30 minutes. And in terms of memory, it's not like super intensive, except for the function that calls remove duplicate function. And now these are plots that are showing comparison between, sorry, it's cut out here. So the y-axis for these plots would be the CellRanger results, and the x-axis are the scPipe results. And again, for the 20% data, so on the left, you will see the counts that are overlapping with the cells that are called from CellRanger and from scPipe. And you can see that the correlation between them is pretty high. And on the right, same for the features, overlapping between CellRanger call and scPipe call cells, which is also pretty high. So, and now this on this Venn plot on the Venn diagram on the left, you will see that CellRanger and scPipe ATAC are mostly calling the same cells. And on the box plot in the middle, this shows that what are the cells that are called by CellRanger, but are they shared with scPipe or not? So red means they're not shared, green or blue means that they're shared. So here, one thing that I noticed was that mitochondrial percentage is actually lower in the cells that are shared with scPipe. So I guess it's a good thing. And on the UMAP, the far right, you see the clustering for this CellRanger cold cells. And they're colored by whether they're shared again with scPipe or not. So the red means that they're not shared, which obviously do fall into like a very confused blob in the middle. But imagine you like take all of these red cells out of this UMAP. It actually do clean the UMAP pretty well, but that's not simple as that, because there will be missing data in this from CellRanger, and I explain that to you in the next slide. So in this, we are looking at this data the other way around. So what are the cells that are called by scPipe, and whether they are shared with CellRanger or not? So here, as you see, the variation in the box plot are not as different as it was in the previous, whether they are shared or not. And on the UMAP, it's, the clustering is like very distinct. And if you see these red cells that are falling within these clusters that are not called by CellRanger, that leads to missing data that I was mentioning in the previous slide. And now is, now here we've done the clustering in Seurat. And then we are looking at, so as I said in the protocol slide, we know the ground truth about this data. So we can label that using those the cell line information. And on the left, you see the clustering colored by the cell line. And then on the right, you see the clusters colored by whatever is identified by Seurat. And we use the ARI, the adjusted rand index, and the nearest mutual index to see the similarity between these clusters. So meaning, as it came up again and again, if you have value closer to 1, that means the clusters are similar between the two. But here, the lower ARI and NMI can be due to Seurat not being able to identify between the some cell lines. For example, this H228 and H1975, these two clusters, Seurat is confused about them. So, but then now again, we are looking at the scPipe results. So scPipe actually can clearly separate the clusters out, and that is reflected by the higher ARI and NMI as well. And so the final feature in the in scPipe is now if you have a barcode match file, meaning that whether you have corresponding barcodes from one SE object and another SE object, you can integrate those in a multi-assay experiment object and take it away from the package to do downstream further analysis or integration using SE-friendly tools. And in my penultimate slide, I want to thank a lot of people, so especially from the Richie lab at WEHI. Original authors of the package, Louis, Xian, and G, and also Xian and Pete for keeping the package alive and maintaining it, and also Jafar and How You for generating results, and also Matt for your patience and for answering my stupid questions. And also, I want to thank the research computing staff of WEHI as well. I don't think they get enough recognition. And I also want to thank my current lab at ARMI, Monash, Rosel Diaz lab, and this work was funded by the generous funds from Chan Zuckerberg Initiative and CSL. So I'll leave this summary and future direction on screen while I'm waiting for your feedback, comments, or questions. Thank you.
Thank you.
Thank you for the good talk. I was wondering what kind of normalization you are using for single cell ATAC-seq.
Oh, we actually don't do normalization. Yeah, we're just doing the pre-processing. Yeah, so just filtering and cleaning it up. So normalization can actually happen downstream with like packages which are specialized. Sorry? Yeah, we are not looking at the counts per se. We are just looking at the reads and yeah, the quality of the reads. So count matrix would be generated, and then that would be an SE object, and that you can use to do the further normalization and things.
Hi, Shani. Great talk.
Thank you.
Two quick questions. One is, how do you do peak calling? Like, which tool do you use, and how do you handle multiple samples? And another question is, if I do like the binning approach that you mentioned, does scPipe use any type of special matrix configuration, like sparse matrix and delayed operations to handle that large matrix that you're going to have from binning counts or binning features, actually?
Oh, okay. So the peak calling is on the bulk level. So not on, it's not discriminated on the sample or anything. So any sort of, so the whole BAM file will be used for the peak calling. So I understand that the sensitivity of the peaks would be low in that case, and that's why we had the binning option. But binning option is, I think it's quite simple. So it's just have like a bed file at the end. So you do the overlap between that bed file and the fragments that we have, and then see what are the overlapping and how many of them are overlapping, and that's how we generate the counts from that. So does that answer your question?
Thank you.
Yeah.
All right, thank you, Shani. Please join me in thanking Shani for her talk.